Okay, so let's talk about cholesterol synthesis. So let's go back and start with our usual story, our usual starting point. So the first thing we did is we took glucose to pyruvate, did our glycolysis, took the pyruvate, put it through the pyruvate dehydrogenase complex, and we got acetyl-CoA. One of the things we can do with acetyl-CoA is if we need energy, we can put the acetyl-CoA through the Krebs cycle, make the stuff that makes ATP, as in our NADH, FADH2, send that to the electron transport chain, and make ATP. So that was story number one that had acetyl-CoA in the mix as part of the story. Of course, we went back and we told our glycogen story, our short-term storage story. We went back then and we told our lipogenesis story, where we made our fatty acids, made our triglycerides out of acetyl-CoA. Of course, we had lipolysis, where we could go the other way and break them down. We also talked about how we could make ketones, ketone bodies, out of extra acetyl-CoA. What we're going to talk about today is another place we can go with acetyl-CoA. Our acetyl-CoA can also go and make us cholesterol. So cholesterol gets important. It's a molecule we need for a number of different things. It's a membrane component. It has a lot to do with the fluidity of our cell membranes. We need it for steroid hormones. We need it for a number of different things, bile salts, vitamin D, things like that. So what we're going to do today is we're going to make that cholesterol. So making cholesterol has a lot of different steps. We're not going to go through every single step. We're going to go through the important ones, and we'll talk about where we're skipping over some steps and some stuff we're doing there. So like we said, we're going to start out with our acetyl-CoA. Another direction, another thing that acetyl-CoA can do. So what we're actually going to do with that acetyl-CoA is take more than one acetyl-CoA and put them together. So this is going to sound a little bit like your ketone body story. So we're going to take the acetyl-CoA, take another acetyl-CoA, and we're going to combine those. We're going to combine them to make acetyl CoA, sounding kind of ketone-ish. I'm going to take one of the CoAs out of the picture. Started out with two CoAs, ended up with one, so we did take one out. Then we're not done. We're going to go and take another acetyl-CoA, add another one in. And as you notice, our carbon chain is growing with this. Acetyl-CoA has two carbons in it, adding two more, adding two more. So this is going to sound a little familiar too. Get rid of another CoA. And we're going to make a molecule called HMG-CoA. I won't write the whole thing out, but it's 3-hydroxy, 3-methyl gluteryl-CoA, or beta-hydroxy, beta-methyl gluteryl-CoA. So most often known, most commonly known as, for that reason, HMG-CoA. So since you're more likely to see HMG-CoA, that's what I want to write here. The enzyme that makes this is HMG-CoA synthase. Pretty descriptive, pretty easy, except it can complicate your life. So the next step with the HMG-CoA, though, is we're going to convert it to a molecule called mevalonate. So that's our next step. This is the step that's probably the most important step in making cholesterol, and it's the most important step for a couple of reasons. One is it's the rate limiting step, really slow step, one of the slowest steps in all of our metabolism. So it's the rate limiting step, and the enzyme that does it catalyzes that reaction is HMG-CoA reductase. Really, really common thing to get asked. So it's an enzyme that I'd make sure I knew. What's the rate limiting enzyme in cholesterol synthesis? HMG-CoA reductase. But you know it's going to be a distractor for that. So be a little careful there. This step is really interesting though. Being the rate limiting step, of course you have to know the enzyme, but this is a step that's modulated or regulated by cholesterol drugs. So you're eating too many double cheeseburgers. Your doctor says, hang on here, you're making too much cholesterol. You need to take Lipitor or another statin or statin. What the statin is doing is modulating this reaction. So that's where this enzyme gets so important, where this step gets so important. So anyway, once we get to mevalonate, what we're going to do is we're going to convert that mevalonate to isoprene units. So isopentyl pyrophosphate first, and then some isoprene units. So let's figure out how to draw this. We'll take our arrow back up. So we're going to go up to isopentyl pyrophosphate, and we've got a whole bunch of molecules here, a whole bunch of units, and a whole bunch of steps. So we're going to convert the isopentyl pyrophosphate to isoprene units. And what we're going to do is we're going to make a whole bunch of those, because what we've done here is we've started out with a 2-carbon molecule, acetyl-CoA, added two more, and we've started building it up that way. Cholesterol is a 27-carbon molecule. So what we need to do is we keep repeating this step over, adding more and more units, building up this molecule. We also have, with the isoprene unit step, I don't know if color really matters. We'll be consistent. We have about 21 steps in here. So depending on how you want to count it out, how you want to divide it, what you want to do, there's 20 or 30 steps in making cholesterol. Not really necessary that you know them all. So we're kind of hitting the important ones. We'll go back and talk about a mnemonic that's helpful with the type of questions you tend to get asked on that. So in those 21-ish steps, depending on how you divide them, you make a molecule called squalene. Squalene. Tomato, whichever way you want to say it. So we take the squalene, we take that to 7-dehydroxy cholesterol. That then gets converted to our cholesterol. And like we said, we have 27 carbons in cholesterol. So it's a big multiple ring structure, 4 rings molecule. So these are the steps we used to get there. Oh, and I forgot an enzyme that shows up once in a while. When we have the 2 acetyl-CoAs and we're making acetoacetyl-CoA, the enzyme there is thiolase. Possibly something you might get asked, maybe not. So we've got our cholesterol now, and we'll talk about it in other videos, what we're going to do with it. But there's a number of directions we can go. It's part of your cell membranes. That story will be about putting cholesterol in with the phospholipids in a cell membrane. So remember when we were talking about making our triglyceride, we had that phosphate on carbon 3 of the glycerol. If you didn't remove that, it became a phospholipid rather than the triglyceride we were trying to make. And the phospholipid is a molecule that you see in cell membranes. Cholesterol is a component of cell membranes. It's a factor in the fluidity of the membrane. Another thing that we can do with our cholesterol is cholesterol plays a big part in making steroid hormones. So that's going to be things like your progesterone, testosterone, estrogen, corticosteroids, things we'll talk about and you'll talk about in other classes. You'll see cholesterol playing into that. Another purpose you're going to see with cholesterol is cholesterol plays a part in making your bile salts. So remember when we were talking about breaking down fats and digestion last term? We said we have these fats, we mix them in, we emulsify them with bile salts. We take the big globs of fat, make them into little droplets. So there's your bile salts coming in. The other thing cholesterol will do is it plays into your lipoprotein story. And we're going to do that in a separate video and talk about lipoproteins and transport. So let's go back, run through the mnemonic quick for making cholesterol and the things you typically get asked on it. So like we said with mnemonics, we do them for two reasons. One, I see no downside to doing them if it is something that might help you. The other thing is very often there's a hole in the mnemonic that will get you in trouble if you don't know to go and look for the hole. So our mnemonic is going to start with A. So that's our acetyl-CoA, our acetyl-CoA. Then we're going to go to our HMG. So our H will be happy. Next thing is our mevalonate, happy man. Next is our I, isopentyl pyrophosphate, or the isoprene units. Really the isoprene units more so. A happy man is squalene, seeing, cholesterol, seeing chickens. And like we said, it's 20 to 30 steps, depending on how you want to divide it. But those are the steps you tend to see most often. So they're probably the most important steps. So we'll stop there with our making cholesterol. Next step we'll go to is about taking that cholesterol, building it into lipoproteins, and transporting it.